Dimohon
Cek.
kembali ke tempat karena acara akan mulai.
Cek, cek.
Cek.
Ladies and gentlemen, welcome back to International Conference on Life Sciences and Biotechnology. The first session is Plenary Session 1. The keynote speaker is Professor Harald Churhausen, Chairman Cancer Research Center, Heidelberg, Germany. The Plenary Session 1 will be conducted by Dr. Renat Kartika Senjarini. Dr. Renat Kartika Senjarini is Head of Biology Department, Faculty of Mathematics and Natural Sciences, Universitas Jember. She is also organizing committee for the AAD alumni in this event. Now, we invite Dr. Renat Kartika Senjarini to come to the stage. Time is yours.
Bapak Ibu sekalian, sebelum saya mulai, mohon perhatiannya agar semua handphone dimatikan atau disilentkan supaya tidak mengganggu jalannya acara. Mohon perhatiannya Bapak Ibu sekali lagi agar handphonenya disilentkan atau dimatikan selama berlangsungnya acara. Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Good morning, the honorable all respected guests and the second ICOLIB participants. This is really a great honor for me to be a moderator in this special keynote speaker season. First of all, please allow me to welcome our keynote speaker, Professor Harald Churhausen, 2008 Nobel Prize winner in Physiology and Medicine, His Excellency Professor Harald Churhausen. Herzlich willkommen. Churhausen has been traveling far away from Germany to Indonesia. Jember especially because Jember is compared to Jakarta and Bandung maybe like a village, but our spirits I think comparable to UI from Jakarta, for example, or ITB from Bandung. So I thank you very much for Pak Hasan in supporting all out to our event, and also to Professor Churhausen, who has come to our university because from all the Nobel Prize, not even contact from all our finger that has come to Indonesia. So this is very rare occasion. Vielen herzlichen Dank. And now, I don't want to waste more time. Let me give a brief introduction. Professor Dr. Harald Churhausen from German Cancer Research Center, Heidelberg, Germany. Professor Churhausen and his team has made a breakthrough in 1982 and 1983 when they were able to isolate HPV-16 and HPV-18 as the virus types responsible for cervical cancer. Based on these findings, vaccine has been developed against cervical cancer, one of the most common form of cancer among women and also in Indonesia. I give now time to Professor Churhausen to deliver his lecture, please.
It's on, yeah?
Kunnen Sie het snel proberen?
Yeah, I think it works. Okay. I think I move a little bit outside, yeah? Well, thank you very much, Katika, for your kind introduction. And, oh, there's the pointer. I forgot the pointer, yeah. Sorry. That's fine. Thank you. I know. Well, thank you very much. Professor Hassan, Rector, Distinguished Rector of the University. It's a pleasure being welcomed here. Dr. Professor Vice Rector Sulfita, thank you. And distinguished colleagues, Dr. Bakeling, members of the German Academic Exchange Service, the DAD in Germany, distinguished colleagues, ladies and gentlemen. What I'm going to do today is to discuss with you some aspects on which we are presently working. When I say we, it's in particular my wife, Esther Michel de Villiers, and also myself, and a group of devoted people whom I will recognize at the end of my talk. Now, since almost prehistorical times, namely starting around 1962, I started to work on viruses in human and infectious agents in human cancer. For quite a while, we worked on Epstein-Barr virus, later on polyoma, adenoviruses, and for about 30 years on papilloma viruses and human cancer, cervical cancer in particular. And at the turn of this century, my wife and I, we both became a little bit tired to talk always about the same subject, and decided to change a little bit the topic and turn to a very different topic. And I will try to explain you a little bit why we did it and what is the reasoning behind it. Well, it doesn't move. No, it's too far. Go back. Okay. All right, this is the slide which I wanted to have. Now, the reason is that we know since a very long period of time that there exists a number of agents in humans which basically do not cause, in the majority of cases, do not cause any disease, but they persist in a latent form for lifetime. Occasionally, under conditions of immunosuppression and certain other conditions, they may be reactivated, they may be produced in larger quantities and excreted, for instance, in the urine or in the saliva, but they have some of them have never been shown to be linked to human cancer in these animals. Although they cannot replicate, they cannot propagate in the same animals. It's in a way an abortive type of infection. Only very specific genes of the genome of those agents are expressed, and they are apparently carcinogenic there. So in a system in which they cannot replicate, they're suddenly carcinogenic. They produce cancer. And in systems where they replicate, in this case in humans, on the right-hand side of the slide, they are non-carcinogenic. So that triggered the idea, is it possible that we have something in some of the animals which are around us, or pets, or farm animals, which they harbor, where the agents replicate in those animals, on the left side of the picture, but do not produce tumors in them. And only in the conditions when they are transmitted to another species, in this case to humans, where they cannot replicate, are they possibly carcinogenic under those circumstances? That was the basic idea behind the story, which I'm going to tell you today. Could you go on with the slide? This doesn't function here. Could you? The slide doesn't move. Okay. I think I missed one in between. Okay, this is all right. Now, we do today is that approximately 21% of the global cancer incidence is linked to infections. Linked does not mean that they are the sole agents for causing cancer, but in addition to some other factors, they may cause cancer. I will come back to this later on. We can roughly calculate that close to 1% of the global cancer incidence is linked to parasitic infections, mainly to schistosoma infections, to liver fluke infections, and very few others. And they are preventable by chemotherapy. I mean, effective chemotherapy is available in order to clear even an infected organism by those agents. We know that about 35% of cancers linked to infection, 35 of those are linked to a bacterial infection. Helicobacter pylori, stomach cancer is a very frequent human cancer, plays here a very significant role. And there are close to 64% which are linked to virus infections. And of those, again, about 60% are preventable by vaccination, and the vaccination provides a long-time resistance against reinfection. Next slide, please. Now, in spite of the large number of cancers which have been totally sequenced by now, since about 2008, there's not a single additional agent which has been linked, at least convincingly, to any kind of human cancer. And this has, of course, a kind of a consequence. Either we know the vast majority of infections which are presently linked to cancers, or the involvement of infectious agents, if they are involved, is following a different pattern from well-established tumorigenic pathogens. And we decided to look into the latter possibility. And I will explain to you why we did this. The one cancer in humans which is known since a very long time to be linked to a nutritional factor, that's cancer of the colon. There are more than 100 follow-up studies which have been conducted in the past over a long period of time, which all revealed that the long-time consumption, as it was assumed, of red meat is a risk factor for this type of cancer. And since countries with the highest rate of red meat consumption, Argentina, Uruguay, New Zealand, USA, most European countries, and interestingly, I will come to this specifically, also Mongolia, which is also linked to higher red meat consumption, but the latter one is quite different from the others, commonly reveal a high rate of colon cancer. For quite a long time, it was quite clear, or it seemed to be quite clear, what is the reason for this high risk. Because if you boil, barbecue, roast, or grill red meat, there's some chemical carcinogens which arise during the kind of preparation steps for this meat consumption. And these are in particular aromatic hydrocarbons and a couple of others, heterocyclic amines, and also nitroso compounds which arise in this process for consumption. And that seemed to make the story clear because obviously those chemicals, if you feed them to rodents, they are tumorigenic under those conditions. Also the doses which are provided to the animals usually are about 10,000-fold higher than the doses which is in a good grilled steak or something like this. But anyway, next slide please. There's a few things which didn't fit into this whole story. Namely, first of all, epidemiological studies did not reveal an increased risk of colon cancer if you consume mainly chicken, poultry, or fish. Fish even is supposed to have a protective effect, and as the group produces the same chemical carcinogens as a good broiled steak, for instance. And that didn't fit into the epidemiological pattern here. And fish even seems to have kind of protective effect under those circumstances. Next slide, please. Now, when you look into the geographic epidemiology of this type of cancer, of colon cancer specifically, then you see that there are a few things which are striking. It seems that everywhere where the Eurasian type of cattle is being kept, this Holstein or Alpine cattle or so, the dairy cattle, the rate of colon cancer seems to be high. Interestingly, in India, India has the lowest rate of colon cancer globally. In India, of course, Hindus don't consume any beef, meat at all, but they also consume relatively little in comparison to other countries of other types of meat at the same time. But the most striking example here is Mongolia. Mongolia, which is supposed to have one of the highest rate of red meat consumption, and yet it's almost the second lowest rate of colon cancer. So how can we explain this? So we looked into the different type of species of cattle which are being kept globally, and it was a bit striking that it seemed that everywhere where preferentially, like in Mongolia, yaks and a different type of cattle is being kept, an indigenous cattle which was mainly crossbred with the Southeast Asian Zebu type of cattle. The rate is low. And also where these Zebu type of cattle are kept besides Southeast Asia, in Bolivia in particular, also the Batusi cattle in Africa, they have a low rate of cancer. So in a way, after a certain period of time, I thought I'd become a specialist on different cattle breeds. And it seemed to be linked to more or less one specific type of cattle. Next slide, please. And it's interesting to look into the time change in colon cancer development. The left part of the slide shows the development in some of the Scandinavian countries. And you can see it here for the period between 1975 and 2010. There's a slight increase of the colon cancer rate in those countries. If you look on the right-hand side of the slide, the situation is very different. First of all, there's one country where the colon cancer rate is going down. That is the United States. Approximately since the year 1990 or a little bit later. And that seems to be due, as it has been described in a couple of publications, due to an intensive colonoscopy program, discovering the precursor lesions of colon cancer, removing them, and getting a reduction in the rate of colon cancer by those circumstances. There's a slight decline also now visible in Canada and even in some of the European countries. Very recently, there's a decline visible. But the most striking examples here are clearly Japan and Korea. Because in Japan, starting approximately since if one interpolates this a little bit, probably since 1965, 1960, there is a rather steep increase in the rate of colon cancer. And in Korea, about 15 or 20 years later, the same happened as well. This is approximately about 15 to 20 years after the Second World War and 15 to 20 years after the Korean War. And you see it on the lower part of the slide that within this period of time, the rate of colon cancer increased dramatically and in conjunction with the assumption of with the consumption of meat during this period of time and reaching in the end in Japan by the year 2000 and also in Korea even more the rate of the high-risk countries in Europe and in North America under those circumstances. India has also a slight increase, but it's by far not the same as it is for colon cancer. So apparently something has happened in those countries within this period of time which increased dramatically the risk for colon cancer. What happened, next slide please. What happened was in Japan as well in Korea, there was a large imports of red meat, in particular from the United States, of beef, and also to some degree of pork in both of these countries, each time after the respective war period subsequently. And within a lag period of something like 15, 20 years or so, the rate of colon cancer started to increase after this period of time. And in particular in Japan, the consumption of raw meat became very popular, shabu-shabu, and also of raw slices of beef became very popular. In Korea, the so-called Korean yakro became very popular. It's a kind of a crude raw tartar of beef. And that seems to be now becoming more and more popular in a couple of additional countries here in Southeast Asia. At least I was told that this is the case right now. Now this slide listed now a careful review of the literature on cancers which have been linked to meat or in part also to milk consumption. And you see here that the evidence for colon cancer is quite high. The consistency of the observation is very high in this case. There's even for breast cancer, and surprisingly too for lung cancer. I come back to this later on. And for prostate cancer, there's a kind of substantial evidence that milk and red meat consumption seem to contribute to the risk of the respective forms of cancer, in particular milk consumption here as well. For the other diseases listed here, the evidence is low and it's not very consistent, but clearly some reports exist even claiming that there is some indication that meat or milk or dairy product consumption plays a significant role under those circumstances. Now, next one, please. So we assumed, in view of the infections which we know to be tumorigenic in humans, that here a similar factor may play a role, which is taken up by us, persists in specific type of cells as a genome, most likely, for very long periods of time. And under those circumstances, usually nothing is going to happen except that some additional events, mutational events or epigenetic events, the methylation of the DNA, would somehow activate this latent factor under those conditions and lead to tumor formation. And as we know it from all the other systems which have been linked to human cancer, none of them is by itself sufficient for cancer induction, but it always needs some kind of interaction, either with chemical carcinogens, physical or biological carcinogens, which by themselves are mutagenic, cause mutations in the same type of DNA, that this type of synergism, and I'm going to talk about this on Wednesday, that this type of synergism might be one of the factors which would eventually lead to cancer. Next one, please. Now, we developed a kind of an hypothetical model of our assumption, namely that this infectious factor is taken up if you take the long line, dark line here, as a life expectancy of humans of about 80 years or so, that within this period of time, the consumption of red meat would lead to the uptake of some potentially carcinogenic infectious factor, which would somehow persist in a latent form in the respective types of cells, and in addition to the later exposure to chemical carcinogens, for instance, by the consumption of cooked or cured red meat, which produces chemical carcinogens, which were discussed earlier, that under those circumstances, an activation would take place, which eventually leads to the precursor lesions of colon cancer and subsequently to colon cancer itself. And that inherited genetic modifications would basically play the same role as those which have been acquired later in life by modifying specific genetic cascades which control these types of persisting infections and which are missing or not functioning due to genetic failure. Next one, please. Next slide. Okay, well, this was the reason for searching for potential agents which come here into question, which would be theoretically of importance. So that was, the idea was to look into the serum of our dairy cattle and also into the milk products of dairy cattle, and to see whether they contain something which could fulfill this type of speculation, which would fit into this picture. And indeed, we came up by now with a large number of relatively specific molecules which we could group. They were all ring molecules, single-stranded ring molecules, which were all grouped into 4 different groups already due to their molecular structure. And they were mainly characterized by Esther Michel, and she will talk about it on Wednesday, so I will not go into the details right now. But the interesting part is that at least those which we labeled as group 1, 2, and 4 contain sequences which are closely related to bacterial plasmids. Now, that of course raised immediately suspicion. We are dealing here with a contamination, of bacterial contamination, which exists in our samples. And I will dwell on this now quite intensively, whether it is bacterial contamination or not. Bacterial sequences have been discovered virtually in every sequenced tumor right now, and are almost uniformly instantly discarded from further analysis because they're considered as contaminants of this material. Well, since we approach this from a different angle, namely from work which we did in our lab, particularly as Michel, on TT viruses, which show a similar structure and are widely spread in humans, virtually every one of us continuously carries some of these types in his or her blood without noticing any kind of pathological symptoms under those conditions. Initially we looked for similar molecules in cattle and found those which are listed here. By now, more than 40 individual types, different types, belonging into one of those 4 groups which we characterized here. Next slide, please. Next one. Okay, before coming to a further analysis, let me make a little detour at this moment and look into another type of cancer which has been linked in part to milk and red meat consumption, that is breast cancer. If you look into the geographic epidemiology of both types of cancers, there are some striking similarities, but also a few discrepancies in the occurrence of these 2 types of patterns. And all these pictures originate from the International Agency for Research on Cancer in Lyon. Basically, the high-risk countries for colon cancer, as showing in the lower part panel of this picture, are to some degree concordant with in the upper part with the high-risk areas for breast cancer as well. Interestingly, Mongolia and also Bolivia are countries for both cancers with a relatively low incidence of both types of cancers. In India, there are some differences because during the past 20 or 30 years, the breast cancer incidence is rather increasing quite substantially in comparison to colon cancer right now. And otherwise, there are a few minor differences which I cannot discuss for time reasons right now. Next slide, please. Now, coming back to the same picture, in part to the same picture which I showed you before on the left-hand side, upper left-hand side of this picture, we compared the colorectal cancer incidence in some of the same countries with the breast cancer incidence, and also the scales are not identical in both of the left and the right-hand side of this picture. It is evident that in Japan and Korea, the breast cancer rate also increased, but not to the same dramatical extent as colon cancer did. And in India, and that is, this slide shows it only up to the year 2005, up to now, it even increased more dramatically in India, the breast cancer incidence there. So something is different in breast cancer in comparison to colon cancer, and we suspected that the low increase in breast cancer in Japan and Korea contrasts the steep increase of colon cancer in the same countries. And so the other speculation was that the target cells for colon cancer can be reached without any kind of immune interference. I mean, the colon cells are directly exposed, so to speak, to the infectious agents which is taken up with the nutrition, whereas in breast cancer, some kind of transport has to occur, either systemically, by a systemic infection spreading via the bloodstream of the respective individuals, or by the transfer of specific types of cells. That specific types of cells may transport these agents to the target sites, in this case, to the breast. And we suspected that the agents linked to both diseases maybe differ from each other, or they could replicate and originate from the same species. Next one, please. So the model for breast cancer, for those reasons, was slightly different from that of colon cancer, namely that we anticipate that something is happening in a very early period of life, namely in the first 2 years of life, probably even more restrictive in the first year of life, by taking something up at that stage which persists, is transferred to some other target organs, but during this course we would anticipate that neutralizing antibodies will develop against the respective type of agents, and that subsequent infections at later stage of life would not have any effect in this case whenever you have a transport through the organism due to the neutralizing antibodies and to the neutralizing effect of those antibodies. And that has otherwise continuously following up their nutritional habits. If this model is true, they would be senseless. Because if you don't include the first period of life, particularly starting already in pregnant women and subsequently during the first year of life, it would be probably senseless, and the money which is being spent for this is enormous and not really usefully applied. Next slide, please. Now, so there's one other interesting result which may point into this type of direction, namely a Swedish group published a short time ago by taking advantage of the Swedish Cancer Registry, which really comprises the whole country, that they identified lactose-intolerant persons, those who do not consume milk or dairy products mainly at all, and compared it to siblings and parents who are lactose-tolerant, who can consume milk and dairy products quite well. And they identified about 23,000 lactose-intolerant Swedes, and compared them to about 70,000 Swedes which are lactose tolerant, siblings or parents of the same ones, so optimal controls. And they noted in the study that there was a decrease in the lactose intolerant people of 21% for breast cancer, and interestingly of 45% for lung cancer. I didn't find any data on the smoking habits of lactose intolerant people, but we assume that it's not very different from the lactose-tolerant ones, but I don't have any data on this line available. But it's clearly an interesting observation. Next slide, please. Now, do we acquire infections from bovine milk consumption? Well, I said before, epidemiological data suggests that events early in life could play a significant role in some cancers, and probably also for some chronic diseases. And now we come to an important point. The prolonged breastfeeding periods of 6 months and more seem to play a very important role for the risk of certain types of cancers and also neurodegenerative diseases. Next slide, please. So it's interesting to look into what is happening by nutrition, by breastfeeding, for, let's say, for persons who are breastfed for babies who are breastfed for 6 months or so, because human milk contains some specific types of sugars that have been characterized by now. Here's the lower part of this picture: fucosylactose 2' and 3' fucosylactose, the allyl lactotetraose, and even interferon gamma. Particularly these sugars, they are almost exclusively present in human milk. They're not present in bovine milk. Bovine milk also contains some sugars, and those types of sugars have a protective effect in the case of bovine milk for infections which affect young cattle shortly after delivery, because these sugars bind to receptors on the cell surface, And this binding blocks the receptors for specific types of infections. In humans, these sugars block, for instance, against noro and rotavirus infections, which are quite frequent in several countries, particularly where the nutrition, where the breastfeeding period is very short and the nutrition is poor at the same time. So it's a protective period, and during this protective period of at least something like 6 months, the immune system of the babies is maturing, and in the weaning period subsequently, when dairy products are somewhat, okay. In addition, then the immune system can more rapidly interfere with those types of infections than in an unprotected baby at an early stage after delivery. And the other interesting aspect, it's a little bit complicated, the story, which occurs subsequently is with the dairy products, we take up some sialine acids, which are not present in human milk and human materials, because we have a kind of a genetic defect. All humans have this defect in the production of a specific sialinic acid, namely the N-glycolyl neuraminic acid, which is present in the animal materials, in animal meat, except for chicken, by the way. And this type of neuraminic acid is being taken up by our nutrition. It's also incorporated into glycoproteins and gangliosides of the cell surface. And under those conditions, these glycoproteins acquire new properties in being receptors for specific type of agents. This is true for polyomavirus, human polyomavirus type 9, and a couple of others, which are suddenly, the babies now become, or the persons become susceptible, who have these modified receptors. So that's an interesting aspect which happens in the weaning period. And we have 2 types of events. First of all, a protective event during a prolonged breastfeeding period for many types of these infections, and subsequently suddenly developing a kind of susceptibility for certain types of infections which didn't exist during the breastfeeding period. Next one, please. So what is the protective effect of prolonged breastfeeding for tumors and other types of diseases? Well, I listed here some of them. I think it's, at this moment, it's relatively good, the protective effect for acute lymphoblastic leukemias, and it's also relatively good for Hodgkin's disease. It's much more poor for neuroblastomas, but they're rare anyway, and not too many reports on this, and also premenopausal breast cancer. It's very difficult, of course, to assess the history of those in terms of breastfeeding when they finally develop breast cancer. But it's moderate for multiple sclerosis, a neurodegenerative disease, and to a certain degree also for diabetes type 1 and 2. Next ones, please. Next slide. So coming back to the story of the isolates which we obtained from milk and from must be here. Yeah, no, okay, it seems to work again. And the interesting part here was that we obtained 2 of these isolates directly from autopsy lesions of a patient with multiple sclerosis, directly from the brain lesions of multiple sclerosis patients. And secondly, also we obtained 2 additional isolates from the serum of patients with multiple sclerosis, not from healthy patients so far. Next one, please. So that initially, due to the fact that these are sequences clearly related to bacterial plasmids, we became interested into the question, are they active in human cells? So we transfected those types of isolated and cloned genomes into human cells, and so very quickly that virtually all of them which have been tested so far are in that sense active, that they induce a different level, and depending on the type, also the RNA of the respective agents, and as we saw later, also proteins of the respective agents. Next one, please. And so looking into multiple sclerosis was quite interesting for us due to the isolates, but there exist a few other data which seem to point that some neurodegenerative diseases also seem to depend on dairy products and on cattle or meat consumption. And the evidence, of course, was quite clear-cut for the bovine transmissible encephalopathy, where you clearly have an infectious agent involved, the cow madness, which was due to the consumption of contaminated food with this agent, a prion, supposedly a prion-linked disease. There's a moderate evidence for Parkinson's disease, that milk consumption plays a role. To some degree also for amyotrophic lateral sclerosis, all neurodegenerative diseases with some kind of inclusion bodies. There's even a bit of evidence for Alzheimer's disease as well, which strengthened our interest in neurodegenerative diseases under those circumstances. Next slide, please. And so for multiple sclerosis, there are 3 risk factors which have been identified in the past and claimed to be the main risk factors for this disease. One is cow milk consumption in childhood, particularly if not pasteurized. Vitamin D deficiency, frequently claimed to be a risk factor because we have a north-south gradient in the northern hemisphere and a south-north gradient in the southern hemisphere for the incidence of multiple sclerosis. Which was linked to vitamin D deficiency, and the reactivation of latent herpes virus infection, so very different from what we have here right now, namely, in particular, Epstein-Barr virus seems to play a significant role. Next one, please. Now, as I said before, we've isolated some of these agents directly from autopsy material of these patients, and we suspected, since many others were negative, that in view of the heterogeneity of the healthy cow blood isolates, remember that I said before that we have more than 40 of individual isolates, that some analogy might exist here, also as we observed it in human papillomavirus infections in cervical cancer, namely suggesting the existence of high-risk types, which are most frequent in these types of cancers, and low-risk types, which only rarely occur in the cancers. Next one, please. Now, what about the virological data which exist? Excuse me. Well, the reactivation, in particular of Epstein-Barr virus, but also of human herpes virus type 6, and the chickenpox virus, varicella zoster virus, has been claimed to be responsible for the initiation of multiple sclerosis. And quite a number of these patients contain antibodies, particularly to Epstein-Barr virus or human herpes virus type 6 in comparison to controls or other neurological diseases. And almost 100% of the multiple sclerosis patients are positive for Epstein-Barr virus. But that is not too surprising because that is a very frequent infection anyway. Next one, please. What about the possible interaction of herpes virus? This is something else. Well, almost a couple of decades ago, during my own period at the University of Freiburg, we studied the interaction of herpes viruses with cells which contained, thank you very much, which contained the DNA of an additional agent, of a small DNA virus, circular DNA viruses like adeno-associated virus, or some of the other single-stranded viruses like TT viruses. And we noted in those instances that the reactivation of a herpes virus in a cell which contains the same other types of agents, here on the left panel of this picture, the small round balls are adeno-associated virus in this case, and the larger ones are adenovirus particles, which do the same as herpes viruses do. This leads to an enormous amplification of the small DNA in the respective cells due to the active reactivation of the herpes virus. A very interesting interaction between two virus types under those circumstances. In the lower panel on the right-hand side, it's shown for TT viruses, where particles are being present visible under conditions of a reactivation of Epstein-Barr virus in one cell. So it seems that cells which are co-infected by two agents, a herpes virus and another small DNA virus, that under those circumstances the small DNA virus is amplified enormously and probably spread to the neighboring tissues. Next one, please. Next slide, please. Now, what about vitamin D deficiency, the other important factor here? Well, again, during the same period when we studied these small particles being amplified under those conditions, a colleague of mine at our place noted that there exists a tissue factor, the tumor growth factor beta, which he called initially the Epstein-Barr virus inducing factor, even prior to the identification of TGF-beta, which is very active in inducing Epstein-Barr virus from a latent form into a reactivated form. And under those conditions, the high level of TGF-beta leads to a high induction rate, a low level just points into the opposite direction. So this seemed to fit very well into the pattern, next slide please, of what we suspected, namely that we could develop a concept of a dual latent infection of specific brain cells by a herpes-type virus, obviously most frequently Epstein-Barr virus, and what we called bovine milk and meat factor, BMMF, as we labeled them. And so the reactivation of the former, the herpes virus, results in an amplification of the latter, spreading this factor to neighboring cells. And due to the expression of foreign antigens in those cells now, which are exposed to the BMMF antigens, a very strong immune reaction with destruction of the affected cells should be the sequence, follow the sequence of these events. Next slide, please. And this is basically a model which was developed here, namely, first of all, that there's one precondition that we deal here with a rare dual infection of human, in this case, of specific human brain cells by a bovine milk and meat factor and a herpes virus, both in a latent form, and that under conditions of vitamin D deficiency, the activation of TGF-beta would occur, and this would lead to EBV reactivation. In fact, there are clinical data which support this, because workers in the Antarctica, which are not exposed to sunlight as a precondition for vitamin D production, that under those conditions they start to shed large quantities of Epstein-Barr virus in the saliva, and upon supplementation with vitamin D, this excretion is drastically decreased under those conditions. That subsequently the induction under vitamin D deficiency of Epstein-Barr virus would lead in a cell concomitantly infected by EBV and the bovine milk and meat factor, lead to an amplification shown here in the middle on the right-hand side of the slide, and the amplification of these other types of particles. And that would result, due to an immune response which should exist in normal individuals, in fact we know by now that this exists in healthy individuals, that this should lead to a rather active response, a focal immune response, destruction of the cells, producing these BMMF proteins in the neighborhood of the cells which start to produce the BMFs, and the onset of disease. On the lower part of the left-hand side, you see an early lesion of multiple sclerosis with a very heavy inflammatory periphery of lymphocytes around the focus in the center, which leads to the destruction of the cells in the center subsequently. And as we can measure right now, also due to the availability now of antibody responses to one group of these antigens, also to an increased antibody titer against what we call the BMMF1 group in these instances. And concomitantly, we observe, as it has been previously observed by others, that there's an indication for EBV reactivation, namely the EBNA1 titer, an indication for reactivation, are consistently elevated in multiple sclerosis sera, which we could also see in our samples. Next slide, please. Interestingly, it's also possible to demonstrate an increase in antibody titers in a different neurological disease against the same antigens, and this is in Parkinson's disease. And you see here on the upper part, on the left-hand side, that there's a significantly higher titer in the Parkinson's disease sera in comparison to healthy controls. By the way, we did in the meantime also age-matched and gender-matched samples, we don't see a difference between age-matched and gender-matched samples, but clearly there is a significant increase. We did not see an increase in the anti-EPNA response, at least not a significant increase, under the same type of conditions, suggesting at least that there's something also interesting happening in the case of this specific neurological disease. Next slide, please. And we do know right now that more than 50% of colon cancer biopsies and colon polyps do contain also Epstein-Barr virus DNA within the inflammatory infiltrates, several of them with some evidence for Epstein-Barr virus reactivation. And there are a number of reports that vitamin D deficiency in patients with pre-neoplastic and neoplastic diseases which have been published recently. Next one, please. I will come to the end very quickly now. This is on the lower part of this picture, a picture from an Italian group who demonstrated the reactivation of EBV in the inflammatory lesion of a colon cancer. In the upper part, there are cells which are expressing a specific type of EBV RNA, Epstein-Barr virus RNA, which is expressed under conditions of latency. Next slide, please. Interestingly, Crohn's disease and colitis ulcerosa, two diseases which bear a higher risk for leading to colon cancer, about a 2 to 3-fold increased risk of colon cancer, show histologically some resemblance to the lesions in this compared again to the multiple sclerosis picture, where you have again also this same type of heavy infiltrate around a focus in the center of the cell. Next slide, please. So basically, we have a model right now for cancers as well. Whether it's true or not, we don't know at this stage. I mean, it's just simply a model. The evidence is poor at this stage as in comparison to the neurodegenerative diseases, namely multiple sclerosis and Parkinson's disease, because first of all, we don't find in the tumor so far evidence, at least for the presence of specific types of these DNAs, at least of the group 1, which has been intensively investigated. What we have not done yet is we have not done an intensive investigation of the 3 other groups which we have isolated from cattle. It remains to be seen what is the outcome. Otherwise, we assume, at least up to this last step, that the situation should be similar to the neurodegenerative diseases, by that again an activation will take place in a later stage, but clearly more data are necessary to prove this point. Next one, please. Well, it's my concluding remarks now. The incidence, the isolated circular single-stranded genomes generally seem to persist as episomes, quite different from a number of viral oncogenic viruses, which commonly, not always, but commonly integrate into host cell DNA. Here we have no evidence for integration. All those which have been tested upon transfection are transcriptionally active in human cells, although depending on the type at different levels. And the groups share substantial sequence homologies with specific Acinetobacter baumannii plasmids, interestingly with very specific bacterial species. Next slide, please. So several of these RNA transcripts are terminally polyadenylated, quite unusual for bacterial plasmids, and provide evidence for splicing events. The adaptation to vertebrate cells is obviously had occurred in the evolution. And in some of the human lines tested, the transfection appears not to result in significant DNA replication of those agents, but specifically in cells of the lymphoid lineage, we have evidence for rather remarkable DNA replication which takes place, and the abortive infection of the small BMMF isolates of a molecular weight, I'm sure that Esther Michel will come back to this later on, with the larger genomes seem to complement each other by co-transfection in the same types of cells, and a high degree of healthy normal individuals contain antibodies to these the major BMF1 protein, the so-called REP protein, in this group, which shows a significant degree of homology among the different isolates. So this clearly shows that infection is taking place in humans to a larger degree, and there are very rarely sera which have been tested, which do not show evidence for antibodies to this antigen. Next slide, please. This is just a replication assay in one of these cells. It was done of one of the colleagues here, which shows the production after transfection on the left panel of with these DNAs. Next slide, please. Not going into the details of this one. Next one, please. So it's interesting to note here that infectious mononucleosis caused by Epstein-Barr virus is repeatedly identified as a risk factor for the development of multiple sclerosis, as a kind of triggering factor in this case, in the early period following Epstein-Barr virus infection, usually 2 or 3 years later on. Small 25 nanometer particles can be seen after EBV reactivation in several Epstein-Barr virus transformed tumor cell lines, and also in Epstein-Barr virus infection in immunosuppressed patients who develop a kind of a hairy leukoplakia at the lateral part of the tongue, and significant antibody levels to the group 1 and the EBNA1, as I said before, in multiple sclerosis patients, and we also see it now increasingly in Parkinson's disease and those conditions. Next slide, please. These are the larger dots here are Epstein-Barr virus particles with the arrows, and you see in the same type of cell these smaller types of particles as in the left-hand slide, but they're all about 25 nanometers in diameter. So that suggests at least that these might be the specific types. We suspect that the receptors which are taken up from or the modification of receptors taken up by feeding babies with cow milk, that they lead to susceptibility for these types of infections. Next slide, please. And so what we can conclude right now is we isolated obviously novel virus-like particle families of bovine meat and milk factors and identified several of these agents as zoonotic infections of humans by antibody responses. In spite of the structural relationship of specific bacterial plasmids, we documented adaptation to genetic activity and DNA replication in human cells. Antibody production in humans demonstrate previous exposure to these infections, and epidemiological and also our serological and molecular analysis suggest this link of such types of infections to neurodegenerative diseases, multiple sclerosis, and also Parkinson's, and possibly also to some human cancers, which is less documented at this stage. So epidemiological data suggests a role, at least in human breast and prostate cancers, and possibly in some other cancers, including, by the way, once again, lung cancer, which I mentioned at one stage. Next slide, please, in addition to smoking. Next one, and that's one of the last ones. Our isolates represent the first example of obviously bacterially derived plasmids adapted to infection of human cells and to replication in human cells. They are very likely to involve at least in one human chronic neurodegenerative disease, but possibly in 2, as I said before. This seems to open a new research field in human chronic diseases at the interface between bacteriology and virology, cancers, neurodegenerative diseases, autoimmune diseases, and possibly even atherosclerosis. Next slide, please. And this is almost my last one. There's only one example which we know in plants, in Agrobacterium tumefaciens, where plasmid is transmitted to plant cells, induces a kind of tumor formation, as shown in the right-hand side of the slide, in plant cells, and a very complicated mechanism. The mechanism is probably not identical to the one which I described here, but this is a really well-established example of a plasmid adapted to being transferred to plant cells, at least in this case. Next slide, please. These are all the co-workers who were actively contributed to this work. And the last slide is now coming here. Next one, please. And these are the external collaborators, which are quite essential for the work which I'm reporting here, particularly from the veterinary faculty of the University of Leipzig. Professor Müller provided us with all these samples of 130 cows, which were quite essential for these types of studies. So thank you all for your attention.
Thank you very much for a very interesting and inspiring lectures. I myself have written many questions actually, but now we have to come to the discussion session. And now I give the first priority to the audience. I will invite 3 participants first for the first session. 1, 2, and 3. Yeah, okay. Bapak yang merah, please give your name and your institution. Thank you.
Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
First of all, I would like to appreciate for the very good presentation and very important topics. Actually, for red meat, we have to consider also how to prepare.
I'm sorry, your identity please, your name
Oh, yeah.
and your institution.
Muslim Suwardi from Universitas Andalas dan Universitas Muhammad Natsir.
Okay.
Well,
Yeah, how to prepare this meat? For example, in Padang we have rendang, the very famous food in the world, not only in Indonesia. Yeah, and maybe for the next research, maybe we have to start. And we know the flavonoid maybe is potential for avoid the cancer. This is my opinion, and I would like to ask you how about your opinion related with how to prepare it and how to cook it. Okay, not only the type of meat or pork or another. Thank you so much.
well, thank you for this question. First of all, we have no real very good data on the thermostability of those agents. We can suspect from the structure of the DNA at this stage that they will most likely be very thermostable. So I cannot predict at this moment whether well cooking of meat would lead to an inactivation of those types of agents. That needs to be tested. We are very intensively trying to develop an infectivity assay for those agents, which would permit really a full testing of those agents. The other aspect which I think is important to emphasize, it's not really related directly to your question, but that is that I do not warn here against the consumption of meat and what is it? Okay, yes. I do not recommend to abstain completely from meat consumption in this case, because I regret to say that almost every one of you sitting here, if you have not been a lifelong vegetarian, has been probably exposed to those agents and will turn out to be antibody positive if we were tested for antibodies, at least against the group 1, which we have so far analyzed. And under those circumstances, I would anticipate that you neutralizing antibodies against those types of agents, and subsequent infection later in life will not play a significant role. But an infection very early at a stage when you did not develop antibodies and when the immune system has not been sufficiently matured, under those circumstances they are probably risky because they lead to the uptake and the persistence. But since we all, including me, are infected by those agents, don't worry much. Your risk will not be bigger if you don't eat meat or milk or drink milk in comparison to those who continue to drink milk and eat beef under those circumstances.
Bagaimana Pak Muslim? How is it? Okay, I think I will go to the second questioner, Pak Adi Adnan from, yeah.
Herr Professor Suhausen, thank you for your interesting lecture. I'm Adnan from Faculty of Pharmacy, Andalus University. I have a question. In India, there are some people group who don't consume any fish in Indian, vegetarian, I mean. What do you think about these people? Are they high risk or low risk on cancer? Thank you very much.
Well, see, the study of vegetarians, for instance, in Europe doesn't make very much sense because most of them develop the habit not to eat meat during puberty or later on, and so that you cannot compare those. In India, the situation is quite different because there exist indeed, obviously, families where the babies already, from the baby age on, there was a total avoidance of the consumption of the respective forms of meat, particularly, of course, of bovine meat. And the rate of these types of cancers probably that is one of the reasons why the rate is the lowest one globally of this cancer in India. You have in India 2 regions where you have a slightly increased evidence for incidence of colorectal cancer. That is in the part of in Kerala, in the southwest of India, and also in the far northeast of India. I learned that in Kerala, up to the recent movement of the government to forbid completely the consumption of beef, that you could even get in a restaurant beef, which was prepared because they have a high proportion in the population of Muslims and of Christians at the same time. So they apparently consume more beef. In the far northeast, the situation is somewhat different because there was an immigration taking part from the Chinese side, and they brought with them the custom of preparing and eating larger amounts of air-dried beef. As a virologist, you would know that air drying is probably one of the best conservation methods for keeping infectious agents alive in those conditions. So it's quite interesting to correlate those 2 types of observations in the far southwest and also in the northeast of the same country. There's a slightly increased rate of colorectal cancer in some of the big cities in India, particularly in New Delhi and in Mumbai at this stage. And that is probably due also to a rather remarkable admixture of other people of different religious backgrounds. Well, people who only eat fish, I mean, there was some data available which showed that consumption, long-term consumption of fish is clearly a protective factor for colorectal cancer, and that the rate is low. And of course, in Japan, for instance, the consumption of fish was quite traditional for a long, long period of time until in the post-war period, in particular after the Second World War. The consumption habits changed, and the introduction of meat, I experienced it myself at several occasions, that you can obtain rare meat at various occasions in the shabu-shabu meals and in the other type. I don't know, forgot at the moment the Japanese name for it, but that you get slices of raw beef as well prepared instead of raw fish in some instances. So that may have contributed greatly to this rather remarkable acceleration of colorectal cancer in this country.
So, Pak Adnan, sudah cukup, Pak? Okay. The next participant is the green, the wear the green hijab. Yes. Please don't forget to mention your name and your institution. Sorry, yeah, Frau Zurhausen, could you please help? I know.
I didn't catch it completely. You are from the Philippines, is that correct?
No. She is from Indonesia.
From Indonesia.
Bu, tolong diberikan mic-nya ke sebelahnya ke ibunya, Bu. Mungkin ibunya mau mencoba
Indonesia has a relatively low risk, in spite of some reports that colon cancer is increasing here.
Okay.
Okay. My wife is responding because she understood it better than I did. We don't understand you. The microphone is not on.
The microphone is on now, I think. Did you say if you eat raw meat, it's like medicine, and you get the bacteria, which is protective? I don't think so. I think, I don't know which bacteria could be protective, but the problem is there are so many bacteria and viruses and everything in the food and everything that we eat, and we still don't know about many of them of what they are doing. So I think you are on the safe side to rather cook and kill them all as far as possible. I don't know whether maybe the DNA would be infectious, we don't know, but it's safer to cook it, I think, unless, for example, with salads or whatever, you wash it very well. But any infected food with bacteria or anything that could be sort of damaging the food as well, I think it may not be safe. And I think with the previous question, the question was with the many spices and the protective effect maybe of broccoli. The problem is we do not know what effect these spices or other things would have on the bacteria or the infectious agents. We still do not know that. But in the end, it's not only one thing that plays a role. If, for example, we took up this infectious agent at some stage, it could be that if we add another bacteria or another spice or whatever, that this extra thing helps the original infection to become dangerous. In other words, or it could also help to protect it. It's like a chain reaction. The one could inhibit the previous thing, the other one may, for example, accelerate it. So it's an interaction between various things, and all these things we still have to find out exactly how it works. But what Professor Zurhausen has been telling us is what we've found so far, and so there's a lot of work to be done to find out exactly how we do it and which one we should exclude and which one we should maybe include. But I think, as far as your question goes, if you eat clean things, and I don't think adding bacteria would be very good for you. For example, Helicobacter causes stomach ulcers, which is not good. So in other words, then you need an antibiotic to kill that. Otherwise, if you have a long-term Helicobacter infection, it could lead to cancer. So it's better to rather kill it by cooking the food than to take it up.
Well, let me add maybe 2 sentences to the first part of the question, namely the bacteria which are present in the intestines. There are 2 types of bacteria, which, by the way, are also, among other properties, they are mutagenic for host cell DNA. That is, for instance, Fusobacterium nucleatum and Streptococcus bovis. And both of these types cause inflammatory events, and due to the activation of the radical oxygen systems, they induce mutations in host cell DNA, so they may interact basically in the same way as chemical carcinogens do under those circumstances.
Yes. I'm sorry because the time is very limited. I'm sure all of us have still so many questions to discuss, but because we have now to move into the next session. I'm very sorry to tell you that we have to close this discussion session. But Professor Churshausen and Frau Professor Churshausen as well is here with us today and until tomorrow, so that maybe if you want to discuss more, you can also do it outside this forum. Thank you very much, Frau Professor Churshausen. So, and now, as a moderator, I thank you very much for all your participants in this special keynote speaker. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Harold Churshausen and Dr. Renat Kartika Senjarini. That was the end of plenary session 1. You may all be seated now.
The second session is plenary session 2. The speaker is Professor Chang Dok-han, Division of Applied Life Science, Plant Molecular Biology, Biotechnology Research Center, Gyeongsang National University, Korea. The plenary session 2 will be conducted by Mr. Suwardianto, PhD. Mr. Suwardianto, PhD, is a lecturer in the Department of Chemistry, Faculty of Mathematics and Natural Sciences, Universitas Jember. His main research interest is in organic chemistry. We invite Mr. Suwardianto, PhD, to come to the stage. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Okay, we continue with the second preliminary session. The session will be delivered by Professor Chang Dok-han. He is a professor in the Department of Biochemistry, Division of Life Sciences, Gyeongsang National University, Korea. Okay. Professor Chang Dok-han, time is yours now.
Okay, thanks. Okay, thank you. My name is Chang Dok-han. I'm really be honored to be invited in this symposiums. Also, I really appreciate it for the hospitalities. I can do okay, thanks. And then I really pleased to introduce my works that in my lab in the last more than 5 years. But I really realized my topic and some slide that I prepared were really specific to, not for the general speakers. But so I'm doing my best to communicate with you about what's works done in the last 5 years. So actually, my field is the plant genetics, and I'm specifically working on the rice, and then working on the genes. So this is topics a little bit complicated, but actually, why we are to introduce the genes involved in determining the plant shapes, plant type, and architecture, especially in the rice. Okay, so. Goals of our labs is try to enhance the yield. So it means in the agriculture, you mean increase the yield. It means that you plant a lot in the same in the given area, and they get the big grains. So this is the very common goals for the breeders and the rich breeders. So. This is the figures how to get the grains yield from the start from the lights in the sun, because it's biomass. It means it's just plant bodies. Is 40% of plant bodies consist of the carbons, and more than 1% is nitrogens. But this carbon, actually, 80–90% of carbon of biomass from the light harvesting stuff. So plants is very important. For, light is very important to plants. Everybody's. And then those biomass, this biomass in the carbon and nitrogen and transport to the grain, and around 10 to 40% in the carbon in the grain you're eating, grain stuffs, is from the biomass. And the other one is, of course, come from the lights. And 79% of nitrogen grain actually transfer from the biomass. So on this scheme, if you see this, it's the light's very important. So improve the breeders. I'm not the breeders, but I'm the geneticist. The breeder try to increase the yield by the target on how to get, how to increase photosynthesis of the area. So this is summary. What? Not going. Okay, sorry. I'm fighting for it not push forward. Yes, no. Probably this. Okay, okay. I can say the next slide. Okay, that's better. But I need the pointers. Okay, next. Okay, I said it next. Okay, next, please. Next slide, please. So this is summary of those from the some publications in the 1940 to between the 1940 to 1980s, and then its grain yield is doubled in the rice and wheat and maize. But if you see if you look at the inside, there was no change in the rates of photosynthesis per unit leaf area. Meaning the plant is to the same capacity of the plant for photosynthesis, but also no increase conversion efficiency of light energy to dry matter. It means plant doesn't change much, but what's the change? Change in the photosynthesis in the aspect, to double the increase of the grain yield was they changed the total photosynthesis per unit land area. It means the plant is more healthier to get the more receive the light and converged in the unit area, and it's called the crop photosynthesis. It's those that these things are happening because improvement of the factors factors like those one, irrigation, you get the more waters and the more fertilizers, and the plant health. This means more get disease resistance stuffs and the durations. A lot of factors that factors can improve that, get the more carbon gains per units, and so you can double them. So this is factors that improved by breeders to get into double the yield. So that's focus that I'm talking about in the rest of the talk is the genes involved and determine the plant type in this canopy architecture. The next slide. This is the cartoon example of the two different types of the plants. You have the one of the open plant types and the other erect phenotypes. So if the open phenotypes, light actually saturated in the top of the leaves, but not much leaf is penetrated. But if the plant is erect, you get the light is get penetrated lower, so sufficient enough to get the photosynthesis. So breeders, of course, the breeders continue to develop this kind of erect phenotypes rather than the open phenotypes. So this is so you can plant more, and then you get the more photosynthesis in the pot plants. So you can get more product of biomass. As a consequence, you get the more grain gain. So the topics that I'm talking about, the genes control this kind of erect phenotypes than open phenotypes. Next slide, please. So we call this kind of shapes of plant architecture determined efficiency canopy photosynthesis. Canopy photosynthesis means you can upper leaf and lower, you can do the photosynthesis same. And then you get the more plant density because narrow plant you can plant more plants and get a more yield. Next slide, please. So in the rice, there are a lot of crop architecture people, geneticists working at plant heights and a lot of other stuffs. But focus on that I'm talking about here is the tiller and the leaf angles. So we want to change the tillers because the rice has a lot of tillers. Also leaf angles. So genes working on the tiller and the leaf angles. So we already previously published that a lot of genes control the tiller and leaf angles of the crop and plants. Next slide. This is a few slides of those previous reports. If there's a wild type genes, it rice, of course. If there was one of the genes mutated, it's called a lazy, it got open up. So it's not a good shapes. So if you look at the gene, they actually in the plant has a hormone called auxins. They do a lot of stuff for doing this kind of open shapes. So if you get this gene is controls the auxins, polarized auxin transport, and then do this kind of, if this gene is missing in the rice, this kind of shapes. Next slide. Also, another gene is we call it, and the TAC1, if they're mutated, this will get a more erect phenotypes. If you see that rice is can plant in like rows, you get a more space here because it's more erect here. So this is genes, if you mutated, get a more narrow shapes of tillers. Next slide. So another hormones in the plant, people using for this kind of plant shapes is a hormones called the brachiosteroid. This is the Finnish groups working on a lot of this. If they knock out, they will knock down the brachiosteroid is missing. So it's same plant, but if you get a brachiosteroid less, this plant is brachiosteroid less. So you get the more shapes of the erect phenotypes. So they using this kind of mutants, they have showing, demonstrate this plant more and get the more yield. The next slide. Okay, so the objective of my lab in the last more than 5 years, trying to find the genes get involved in the plant types, and then we want to look at the functions, and of course, you test for the agriculture utilities for those. Okay, so we're getting more specific, so I'm trying not to specific in this. Okay, next slide. So we get the genes first, and then this is the genes we called it an original sativa modified plant type 1. And this gene was then, here's the exon, and then a lot of transposon inserted. That originally we developed a lot of population of transposon stuff. So anyway, so this is a mutation. If they get into transposon into the genes and they get no expression, and then if you're going to the next slide. So also when you plant them, we have that gene rice and Arabidopsis has the same similar genes. So the basically is, okay, this is a genes in proteins get into the nuclei and they get bind the DNA. So we call the transcript factors. So this is DNA binding protein, and this is the sort of a C-terminus doing the activation or depression regions. Go next slide. So this is actually demonstrated, but you can clear. So you see the nucleus, this protein is getting to nuclei. And the next slide. Okay, this gene is expression. This is Gauss pattern. The blue color is the where this gene is expressed. This is where you're doing to detect expressions. So this is, if you look at the lip, this lip has a lip blade and lip sheath, But in the middle, there's a lot we call the lamina joint. That's strong expression of this gene over here. And then if you look at the tiller area, you can get some specific area. This is area plant response gravities. We call the pulvinar tissue. I'm not going into details. Next slide. So if the plant is get responded gravities, but not every place. So we call the pulvinous tissues. The P stands for P. This is the area in the rice has the responded gravities, and then his gravity. So just to introduce to someone not really familiar with the plants, there are some areas response gravity. So if the plant fall down and rise up after couple of days, so you get this is the area to get actual respond gravity. Next slide, please. Okay, so we just introduced the genes MPT1. So I want to show you the function of the phenotype function, what's the do in the plant type. And the next slide showing is the lip lamina joint. Next slide, please. So this is the phenotype. So we had this is genes mutated mutations. You see that this is the regularized, and then the part, and then you can mutate it. This is open. So its tiller angle is wider, If you look at this is the mutants, and this is wild type. It's lip angles wild, it's more open. So if this mutated teloid angles are higher and they're wider, and then lip angles is wider. If you're going to this is mutation. So if this gene is knocked out, if it overexpressed by the agrobacteria, Dr. Hausen introduced it. Next slide show that if the overexpressed means this is the way the gene is increased. This gene is increased, and then this plant arises, getting more narrow tillers and then narrow lip angles, and then later just too compact to use it. So we did this kind of stuff. So this gene is, if you mutate it and then open it, and then you get the more expressed, you get the more narrow phenotypes, what we want to do. Next slide, sure. So also that's gravity, that I'm talking about gravities. So if that's growing rice, this is rice seedlings, they lay down here and they let them lay down, and they stand up layers. So, but this way, the speed of the stand up is much slower in the knockout gene if this gene is not present in the rice. Next slide is showing the other round. So if this gene is overexpressed, this is more than expressed more than what the wild types, regular plant, they get the faster response for the gravity. So they respond very fast and faster stand ups when you apply the gravities. Next slide, please. So this is showing a phenotypes. Okay, the leaf is wider and narrower. We can manipulate this one. So we did the molecular. This is the quite specific data. But anyway, I have to go through these slides. So we tested for the MPT. You got to clone the gene, and the genetics, the molecular biology, do the function of the gene and the gene action. So do a lot of techniques. So that's when I have to go through it. So I'm sorry about, but I'm trying to get really communicated with you. So first with the laminar joint, between the lip angles determined by the laminar joint. This is the well known. This is the very sensitive to oxygen. If you go into the next slide, this is quite 1965 publication is one of the Japanese groups. It's cut down the leaves, the rice, and they put into the waters with has more oxygen. So if there are a couple of days later, you can see oxygen respond on this laminar joint. This is the way they determine the angles of the leaves. So this is the areas to very sensitive to oxygen. So we want to test how they affect this gene to the oxygen sensitivity. So let's go next slide. So we did the same experiment. This is like, botany schools, botany classes, just adding oxygen to the ethanol and the drop and rolling down to the here, and the oxygen did something to here. And then 2 days later, you can measure the angles. The angles is open in 2 days. So this is the way we assay the oxygen sensitivities, hormone oxygen sensitivity in the plant rice. So next slide. So if the mutants, this is the mutants, okay, if we let the different concentration of oxygen, you see that mutants is very sensitive to wide open oxygen. You can wide open, but overexpressed, this gene doesn't respond to much of the oxygen. So this gene has changed the sensitivity of the plant cell to respond to the oxygen hormones to get. So this is the way. Also, okay. I do not go into detail for this, but this is we treated the sensitivity of the mutants, of oxygen mutants, is inhibited by inhibitor brachyl, another hormone, the brachyl steroid biosynthesis. So if we inhibit another hormone called the brachyl steroid, and that changed the oxygen sensitivities. So it's the two brachyl steroid and oxygen, and they interact each other to get determined the lip angles. Going to the next slide. It's a long story. This is the whole complicated pathway for the brachyl steroid. Okay, going to the next slide. So this is our, after a lot of experiments, we have that how to determine the lip angles by oxygen and the brachyl steroid. We found that there are two different pathways, oxygen and the brachyl steroid receptor. This is the oxygen and brachyl receptor, this oxygen, and this is the brachyl compound. And then there are two pathways, one pathway and two pathways. But we found that MPT1, that gene that I introduced here, was a blocked one pathway, so it get expand. So that's the way they control the MPT1. So this is the sort of, people, usually molecular biologists clone the gene and then analyze the actions. This is part of that process of those, how the actual MPT1s control the lip angles. Okay, next slide. So next one is more complicated. So it's, this is MPT1, actually, and the nuclei, they has to control the expression of a target genes. So mission of the next, our next mission was, this is guys doing in the nucleus, and they probably controlled some target genes, and then that let them do target genes do the control the plant type. So at the same time, the MPT1 could be the impact to some of the proteins. So we have to find this gene, and then try and figure out what's going on in the lip angles and at the molecular levels. And the next was experiments. So we did what the experiments was. We trying to find MPT1 targeting and a lot of molecular techniques, and also we're trying to find the protein intact with the MPT1 protein in nucleus by different a lot of techniques. So if going to the next slide, so we got the RNAs. You have to get the RNA to find the target genes. So we get the RNA from this part of the plants and this part of plant because where this gene is expressed. So we get expressed, and then next slide has to go through a lot of basic molecular techniques to try find the genes. And basically, quickly explain, if you compare the expression in the MPT1 knockout mutants and the overexpression lines, and compare any gene is altered by expression in the mutants or overexpression line and select it, and then we will look at the promoters where the this binding, this is the sequence of this MPT1 bind, where any of the genes contain this MPT1 binding motif in promoters. And then we select the gene, and then we did the in vivo test for the QRT-PCR enzyme chip. This is the whole detail of the molecular techniques. And then finally, we select the genes and then see the phenotype again, whether this is really has to do with the phenotypes of the plant types. Okay, next slide. So we selected a few of the genes, found out that those has to relate to the auxins that we're talking about, plant hormones. So this is a whole bunch of the genes actually targeted by controlled by these genes, and this gene is the function of this gene is related to hormones called auxins. So this is all the transport. And next slide showing another. Except that, and we also other targets in found. The yellow one is the receptor kinase we found on an located receptor kinase, which are showing the pursuit at this gene. What's the phenotype? We just look at the phenotypes. Next slide, please. So this is the target, one of the targets. What you mean target genes? There are genes. This guy is one of the rice gene, and this ATG, and then there are promoters, and the P1, P2, P, it means the putative MPT1 binding site there. So we just directly test whether, sorry, go back. Okay, this is, so, and P1, P2, 3, and this is actually chromatin chip assay. So actually MPT1 strongly bind this side. So we know that this gene is actually controlled by this gene. And next slide showing was this nature of this gene was the protein kinase domain and receptor transmembrane domain and molectin-like protein. This is the one of the carbohydrate binding site, this kind of stuff. So we try the phenotypes get. Genetics, once we get the gene, when genetics get a gene, so they want to look at the mutants phenotype. So that's the way they studying for the genetics. So this is we overexpress, the plant is getting open tillers, so we have the right tracks for this. Okay, you go too fast. Okay, so if this is the rice, it's cut the stem, and they look at the top, and then you can see the tillers open. So if this gene is to delete the kinase, so the domain and overexpress together, get changing for the phenotypes. Okay, next slide. So this is the target genes. So now we have just looking for the interacting genes. So proteins that interact with MPT1 protein in the nucleus. and then see how they're doing. So this is all a scheme to try to understand the function of those genes. So there are a lot of molecular techniques to can, if you're the molecular geneticist, you have to go through the whole different techniques. So, sorry, this is sometimes work, sometimes doesn't. Okay, sorry, I'll be careful. So yeast to hybrid screen, the first we have to, using a yeast to try and find any proteins that bind to MPT1. And then you can express this in the plant cell, in the rice cell, and they see this one is really expressed, interact each other, called biaphysis. And then we also express the protein in it, and they bind each other in vitro, in a test tube, appendal. And then we select those genes, and then look at the phenotype, see how it works. So going to the next step. So next slide. We did a lot of, this is spent 2 to 3 years to get, but we have to end up the 3 genes and actually interact with this MPT1 in the nucleus. So one of the genes is ID13, that's homologous to the MPT1. The other one is kinase in like proteins. But this protein, when you make a mutant, phenotype is very unstable. So students just get difficult working with the unstable phenotype mutants. So we drop out of this one. And the other is keratin-associated protein and the trimeric beta-subunit trimeric G protein. So there are 3 genes. We focus on the 2 genes. Let's go next slide. I'll go speed. This is the technique. So you get 2 genes expressed same time, and if they interact each other, binding each other, you get the color in the nucleus. And then this is the way, and then you can express proteins in the bacteria mixed together, and then you can detect the antibody again. This is still the same complex. So this is the way you're doing. And then next slide. So to get the phenotypes, we had expressed a lot of mutants, these RNA island mutants and overexpression line, and the next slide showing a phenotype. When you RNA island, there are no phenotypes. But if you overexpress, next slide, you see that ID13 overexpress get more erect phenotypes for this. So we are thinking we have the right tracks on this, and now we're trying to get this gene and ID13 independently has showing a erect phenotype, but we're trying to make it both expressed and then see how the phenotypes change. I'm sorry. Go back, please. Can you go back one more slide? So overexpress 13 and MPT1 independently, they has a erect phenotype overexpressed. But if we overexpress both in the same cell, same plant, how they hopefully gets better performance for this. So that's what we're doing. Next slide, the other genes, they're talking about keratin. But this is actually keratin, but it's turned out to be later. It's a trimeric G protein. We get the mutants and tDNA knockout and the phenotypes like this. And going to the next slide. And then this is also test for the 2 gene expressed. At the same time, you get the intact in the nuclei, the 2 gene, and then if the complex make it 2 antibody independent. This is complex and the different separate in the gel and the different antibody can show this is the way to test for the interaction. But when, okay. So when you actually look at this gene, actually published it, and then in the China independently, we published this gene, and this gene is increased. Actually, this is the gene is the QTL gene, is actually also plant type, plant architectures. So the Chinese group has published this gene, and they changed the plant architecture in this specific panicle. So if they overexpress it, that has a 1,000 color weight is increased. The overexpressed it can increase. So we get the we get the fit together. So with the genes we get fish out from the library, they also give the same similar plant types expression. It's going next. So this is actually we overexpressed gene, and then this is a higher increase the kernel weights. So it's good. So we get the good phenotypes of plant type and the good panicles. It's immune indication. So next slide showing also we do the same thing. If what happens if the overexpress both genes, keratin and the MPT1 in the same cell? So we try overexpressed and they make it cross and they make a double combination of those genes, see how they work. So this is what we're doing now. The next slide. So what about the field? This is all the laboratory works. So if they're going out of the field, what it looks like. So that's where I'm showing a little bit the data for this. So we expressed this one, 2 different promoters. One is a ubiquitin, and the other one is a protocol like promoter, and there are 6 lines of the other one. And next slide showing for that in the field, One is the UNEJ. This is ubiquitin promoter. You can see that I show already, but in the field I can show the field test. You see this is the MPT1. This is the parental line. You see the Zeponica and then adding this gene to it, and then you can see the clearly showing this erect phenotypes. If you go next slide. Next slide, please. This is the 6 difference. You see that this is parental line, and if adding a gene by overexpressing a little bit of different promoter, you can see the whole different spectrum of the really narrowing, and so it means you can plant more, basically. So we had the growing more. So this is the one of the. Okay, can go back. So this is the little longer. This is maybe several years ago. We rice in the Korea. They plant in their standard density, so they plant them 30 centimeter by 15 centimeter. That's the way the rice breeder is growing. So this is the way they're growing. This is the wild type. This is the reference, a good elite line. This is also elite line. But if the overexpression line, if the standard density, so if you're growing a regulars, you get a lot of space to thin the lanes. But if they crowded it, and if we deliberately high density, so it doubled the planting. So 15 by 15, you didn't really see the rows, any space in the field. But if you still, this guy has see the space for this. So if you pack them, but still see it, the rows and space, so it's good for the plant. But problem is, if we go into the older, this is the older lodging, this is wild type plants. So regular plant, if you're growing, this is both in the high density. So wild type plant is just lodging it because too crowded. So you cannot using this. But if the overexpression line is still standard, and then you can grow in the field. So if you implement the pact, you can still harvest them in the double space. If you see the still strong, but with the same spaces in the sorry. So same spaces of the wild type, you cannot stand it because this guy's too open, too crowded. Okay, next slide. So but problem is, we just look at very close. The problem is this overexpression line. To be honest, the problem is the decrease of the grain weights, but we don't know why. But we're trying to searching hard. Some of them is really lowers grain weight. Lights decrease the grain weights. So that's what we're trying to figure out. That we're trying to figure out, we selected a better one, or do you remember that one of the genes increased the grain weights, so that we make a double combination with those genes. Okay, that's good. So what we're doing was, we tried to complement the defect, one of the bad characters by like keratin. This is increase the yield, increase the grain. This is increase the plant type, and if they both make, we try and make a both express combination, see how that works. Next slide, please. Okay, so when I look at, I showed one of the breeders. The breeder suggested to me, why don't you bring this gene into another genes, another variety of osmotic 677 Kiwan's Lee was one of my colleagues. Try to introduce. If you look at the next slide, this is the one of. Can you go back, please? Okay, go back one more time. Can you go back one more slide? Okay, no, one more slide. Osman 6 was around, this one of elite liners. 70% rice variety in Turkey is planted on this Osman 6, 97. Okay, go back, open and loose. So he trying to get this gene back to this background, and they get close up and the better shapes. So next slide, he did a lot of crossing stuff to get the Osman 697 to get a better shapes of plants. Okay, so next slide was, what about if this gene is put into other crops, like soybean and lettuce, and then see how they works. Because, of course, there are some soybean, also lettuce has its own homologous MPT1, but in this case, I directly adding a rice MPT1 into the soybean and the lettuce, and then see how it looks like. And then next slide is showing, this is the collaboration with Syngenta in China. They make constructs, and the rice, and put them in the soybean, and they show the slide. And next was, actually, this is the parental lines of soybeans. I really didn't know. I didn't know of soybean. But this is without the tDNA line. this is with TDL line. So soybean is getting, so they send me the small slim nib and erect branches and the ribs and the compact plant architecture. So it means it's starting getting works, but not really fantastic. But next slide was also we collaborate with the seed company, and my friend is working, trying if there's lettuce also put the, leaf is more, lettuce leaf is more just flat, and then they're touching the ground and they get the more disease from each other, from the soil, and so try and put them into the more standard, and then